If you need help learning about cells, this video covers a simple overview of the cell history, the plasma membrane, endocytosis, and exocytosis. Before discussing cellular structure and components, it is important to get a brief history on the discovery of cells. Robert Hooke discovered cells in the 1600s when examining cork under a light microscope. He saw thousands of empty compartments and named them cells. Now our knowledge of cells has improved substantially. As a brief review from previous material, cells are the smallest functional unit of an organism. Cells contain organelles. Cells working together with a similar purpose make up a tissue. The levels of organization expanding from tissues go through organs, organ systems, and finally, organism. Cell theory contains three main points. They are: a cell is the smallest structural unit that performs all vital processes. Cells are the building blocks of all plants and animals. And all cells come from pre-existing cells. Each cell has a semi-permeable plasma membrane that surrounds and protects the cell. The term semi-permeable indicates that the membrane is selective in allowing certain ions or molecules to move past and go through the membrane, but preventing others from entering. It will separate the cell from extracellular fluid, which is fluid that is outside of the cell. The plasma membrane controls what enters and leaves the cell. It is comprised of a phospholipid bilayer that has hydrophilic, water-loving heads, and hydrophobic, water-fearing tails. In addition to the phospholipid bilayer, the plasma membrane will include cholesterol and proteins that are embedded within the bilayer. Cholesterol assists in maintaining membrane fluidity and is colored orange in the diagram. Membrane proteins can be divided into two categories: integral proteins, which span the entire length of the phospholipid bilayer, and peripheral proteins, which are present on either the inner or outer portion of the phospholipid bilayer. Proteins are colored in green. Glycoproteins and glycolipids each contain a carbohydrate component. These specialized molecules act as receptors and aid in cellular communication. The carbohydrate component of both glycoproteins and glycolipids can be seen in yellow. Endocytosis brings extracellular materials into the cell by utilizing a vesicle, which is a membrane-bound sac. The vesicle is gradually formed around the desired extracellular materials by the cell's plasma membrane. Once the plasma membrane has completely enclosed the materials in the vesicle, it is allowed to enter into the cell. There are two types of endocytosis. Penocytosis is utilized to bring in smaller molecules and can be described as cell drinking. Phagocytosis is utilized to bring in larger molecules and can be described as cell eating. Exocytosis is the opposite of endocytosis. In exocytosis, an intracellular vesicle will merge into the cell membrane and gradually evolve to become part of the cell membrane and expel the materials within the vesicle into the extracellular fluid. Both endocytosis and exocytosis are constantly occurring in cells, which means the plasma membrane and vesicles are constantly being recycled and reused. I hope you found this video helpful. Please feel free to subscribe to view additional simple anatomy overviews.